Good evening, everyone. Guten Abend. Good evening, everyone. A very warm welcome to our fellows, partners, former fellows. Welcome back to you, guest speakers, discussants of tomorrow's workshop, friends and followers of the Wissenschaftskolleg, and a particularly warm welcome to those who made it to Grunewald from abroad, despite the very unfortunate travel conditions. So, Observatory, for those of you who don't know the format yet, Observatory is an annual series here at the Wissenschaftskolleg. We established a couple of years ago, five years ago. And each year we have a closer look at one broader disciplinary field, asking what are the major challenges, what are the major transformations, are there significant methodological or theoretical shifts, are there new camps coming up, and so on and so forth. For example, former disciplinary fields we observed were literary studies, theology, law, and sociology. So to this end, we bring together current BICO fellows, former fellows, and a couple of distinguished guests from the field. So, and we always start with a public opening event like this one, which will be followed by a more internal, smaller workshop tomorrow and the day after tomorrow. And those of you who would like to come to this workshop tomorrow, you are all welcome as well. This year, as you all know, the field we want to observe is evolutionary biology. And what we want to thematize is that evolutionary biology, like many other academic fields, is currently undergoing a revolutionary change due to the methodological potential of artificial intelligence and big data mining. Biologists, as I learned from Dieter, are experiencing a kind of gold rush. And the question is what that means for old questions to be answered and new questions to be asked. We are convinced that WICO, the Wissenschaftskolleg, is well suited to discuss that because, as most of you know, biology has been very prominently represented here from the beginning, as far as I know. This is first and foremost due to our permanent fellows from the field. Rüdiger Wehner, first of all, Paul Schmidt-Hempel, Raghavendra Gadakar, and the current permanent fellow Giovanni Galizia, and last not least, Dieter Ebert. Dieter had the initial idea for to this workshop and has been organizing the workshop. Dieter, for those of you who may not know him, holds the chair of zoology and evolutionary biology at the University of Basel, and his main field of research is the relationship between parasites and hosts, their interaction, their ecology, their genetics, their co-evolution. His favorite species being, by the way, the water flea. You may have wondered what kind of poster this is. This is Dieter's favorite species in a kind of Mondrian-esque image. And some of you may remember Dieter's amazing evening lecture a couple of months ago on this topic of parasite-host co-evolution. Dieter was a permanent fellow for 10 years, and he just finished his final term. So this conference is also kind of farewell gift to us, Dieter gives us, and I can only express our gratitude for your engagement for all of these years. Dieter will have the microphone in a minute, but I would like to say that we are proud, thanks to Dieter's and his permanent fellows' continuing engagement, we have been hosting some of the most distinguished scientists of the field over the years. And this is also true for this academic year. I'm particularly grateful that two of our current fellows, namely Leonie Moyle and Matthew Hahn, both from Indiana University in Bloomington, supported Dieter in gathering this excellent group of colleagues for this panel and for tomorrow, and they will also moderate 2 of the panels tomorrow. But I don't want to anticipate Dieter's introduction of these colleagues. Before I pass over to Dieter and the panelists, I have one announcement to make. I would like to direct your attention to our annual Ernst Mayr lecture in the field of life sciences, which some of you may know, organized together in cooperation with the Berlin-Brandenburg Academy of Sciences. And this year, our current fellow, Pablo Marquette, from Santa Fe Institute in Chile, will give the Ernst Mayr lecture. And he will raise the very fundamental question of understanding the phenomenon of life in its diversity as a central challenge in science and philosophy. So a very, very fundamental topic, and I warmly invite you to this Ernst Mayr lecture taking place on the 28th of April, as always in the Academy building on Gendarmenmarkt. And so save the date, 28th of April, Pablo Marquette. Now it's Dieter's turn. The only thing left for me to do is wishing us all an insightful evening and tomorrow an interesting workshop. Thanks.
Thank you very much, Barbara. Dear Vico Fellows, dear evolutionary biologists, friends, colleagues, ladies and gentlemen, it's very exciting for me to be now here, this kickoff meeting. We were waiting for it for quite a while. It's amazing that it now takes place, and it's really beautiful to see that the room is packed full. This is very nice. And I want to say immediately that all of what is happening tonight and in the next 2 days is not only what I have done, even though Barbara put me in the foreground. There are many, many important people, Matthew Hahn and Leonie Meudel already mentioned, but then also the staff of the Vico who kind of tremendously supported all of this. And, I don't see all of you here, but thank you to everybody for this. Science is moving forward in accelerating speed, and sometimes the speed is so high that we may experience it as a revolution. This is where the title of this conference comes from. While this possibly has many reasons why this is happening, 2 of them play a particular role in the field of evolutionary biology, and this is already mentioned by Barbara, big data and artificial intelligence. Big data is actually something which is not so new. Already 50 years ago, there were hundreds of weather stations all over the world, and where people were collecting big data on weather stations. But this has accelerated. Nowadays, we can do remote sensing with satellites. You can place a camera somewhere, and the camera is recording everything what is moving there. You can put entire forests under a network of microphones. Everybody who sequences whatever gene somewhere enters the data to gene bank or equivalent data repositories. Even citizen science is producing big data. If you think about iNaturalist or eBird, where daily thousands and thousands of people report all types of things they are observing in nature. And it's really quite amazing what data are out there. And they are now, and this is a difference to former times, they're now a mouse click away. So each of us can typically rather easily have access to this data and can do something with it. And if the data are not there for your project, it's nowadays so easy to produce big data for your very project just by using cameras, using microphones, using logging stations of whatever sort, or having next generation sequencers to sequence your own genomes. It's really amazing what can be done there. Now this data so far had been analyzed with kind of rather crude statistical methods where you would look how one variable relates to another variable, maybe a little bit more complicated than this, but not really going into the deep, complex nature of many of this data. For this, AI needed to arrive. Artificial intelligence rocks the planet. It's here, it's changing everything what we are doing. And it's possibly the most discussed topic, at least on the university level. I hear it everywhere, constantly talking about it. People use it in various forms, either just as a helping tool, even a spell checker might have AI behind it, or really to do very sophisticated analysis of complex behaviors, for example, to study the evolution of communication, languages of not only humans but also animals, how bees or how great tits communicate with each other. Many, many places where we can really dig deep and understand levels on a complexity which was just unthinkable a decade ago. It's really a quantum leap. It's a revolution, and part of the field addresses questions in evolutionary biology. These are the revolutions in evolution. Of course, there are also voice. You hear quite a lot of people who start to express their concerns. My students ask me, why do we need to learn all of this anyway? I will be jobless in a few years because AI is doing everything. Is AI creative? Can AI come up with new concepts? Where are the limits? What can we do? There's a lot of this discussion, and you see that the spectrum goes from people really enjoying the benefits of it to people being totally worried and having sleepless nights. I hope none of you will have sleepless nights, but it's meaningful to ask questions and to discuss them. And the Observatorium is one way or one place where you can do so. Tonight, here in this session with this panel, we want to discuss some of the questions which we can address with AI and also what we may not be able to address with AI. And so we have this kind of format where we have first the panel discussing among each other, and then we open to the audience, and all of you can contribute to the discussion. I want to introduce you to Adam Rutherford. He's the moderator of the session, running it. He's a lecturer at the University College of London. He's a geneticist and science communicator. He has been working for various important outlets, if you want. So Nature, for example, he's writing for The Guardian, he's producing podcasts, he's writing many articles, and in particular he wrote a number of books. For example, A Brief History of Everyone Who Ever Lived. So now you see, this is your chance. You feature in this book. And this is coming in a new edition. This was published 10 years ago. It's coming now in a new edition. So I make a little bit advertisement for the book. And it's largely expanded, more material, interesting things. Maybe a bit of AI in there. No? So have a look out for this. He also wrote a book called Creation. He wrote another book called How to Argue with a Racist. So diverse topics, roughly all related to history, development ideas in relation to evolution and genetics. And I think I said enough. I hand over to Adam. Please.
Thank you, Dieter. I think I'm going to employ this man to do all of my PR from now on. I was lecturing in New Zealand a couple of months ago, where it's traditional and respectful to introduce a lecture with a Maori greeting, and I think that's a respectful thing to do. Aber mein Deutsch ist nicht besonders gut, und das ist auch schon alles, was ich sagen kann. Also für den Rest des Abends, I'll be speaking in English. So yes, I'm Adam Rutherford. As Dieter said, I'm a lecturer in genetics at UCL. I specialize in the history of genetics, the history of evolutionary thought, and the public understanding of evolution. As a result of studying the history of our fields, I spend a lot of time in Berlin, but not actually in Berlin. So it's a real pleasure to be here in this amazing institution, this school. I think Berlin is my second favorite city on earth. I'm going to start with a couple of quotations, statements really. I'm going to test the panel on this. So, nothing in biology makes sense except in the light of evolution. Very good. Theodosius Dobzhansky. Hannah's one up. The second one is, evolution by natural selection is pretty much the best idea that anyone ever had. That's me.
-1.
You lost the point, yeah.
Then you lose a point. No, I didn't expect you to know that. So as Dieter mentioned, this is how do you pronounce it? Observatorium. Yeah, okay. Is the beginning, this public event is the beginning of a 2-day conference where people will be discussing the revolution, the ongoing revolution, the next wave of evolutionary genetics, and our ongoing attempts to understand the diversity of life on Earth. Now, we are scientists, there's many scientists in the audience, and the currency by which we understand the natural world is data. As Dieter alluded to, we're in an era of big data, more data than we can possibly cope with. The genome in my own field of genetics, the human genome is probably the most complex data set in the known universe. We have supposedly completed the human genome with the Human Genome Project announcing the first draft in July 2000 and then publishing it in 2001, and then the next draft was in 2002, and then 3 and 7 and 11. So the complete human genome has been published about 18 times over the last 20 years, and I think it's fair to say that we don't understand how most of it works now that we have it all. And now we're capable of extracting DNA from people who've been dead for thousands, tens of thousands of years. We can, it's trivially easy to sequence and read and manipulate and edit. We have gene editing experiments done at school age, which were inconceivable 20 years, 30 years ago. And we can use these technologies to cure diseases. The first successful gene therapies after 40 or 50 years of talking about it really came online within the last year. When I wrote that book 10 years ago, I said the number of diseases cured as a result of gene therapy was 0. And that was true at the time. And now the number is about 8. And that is miraculous. So much of the meeting is going to be about genetics and the extraordinary amount of data available to us, the genome being the definition of big data. But studying animal behavior is really at the root of evolutionary thought. It's where Darwin started out on his pathway to understanding the diversity of life on Earth and coming up with his theory of natural selection. So in this event tonight, we're going to be talking about the cutting edge of using AI, using new technologies to ask basically the same questions that Charles Darwin did 160 more years ago, with technologies which were not inconceivable in his era, they were inconceivable when I was an undergraduate. And that's really, really exciting. That's the revolution that Dieter's referring to. We're going to be seeing how AI is helping us to decode the 4 billion year head start that life has on us trying to understand it. So we've got 3 amazing scientists all working at the cutting edge. So I'm going to do in order from left to right here. First, Hannah Kocher is an Alexander von Humboldt professor at Johannes Gutenberg University in Mainz. So you're an evolutionary ecologist trying to understand diversity in evolved solutions to life's problems. So your topics, Hannah's topics over the years, very diverse range from sexual reproduction to aging, dispersal to migration, and social evolution. And Hannah was a Vico Fellow here in 2013. And then next to her is Tim Landgraf, Professor of Artificial and Collective Intelligence at the Dahlem Center for Machine Learning and Robotics. That's the Freien University.
Yes.
Yes. A computer scientist whose work in AI and robotics we're going to see in a few minutes as powerful analytical tools and providing new lenses for asking questions in evolution, ecology, and behavior. And on the far left, I mean physically, not politically, actually I don't know, is Sylvia Kramer, who's a professor at the Institute of Science and Technology, Austria, ISTA, and she's a behavioral ecologist. Ants are her thing, which we're going to see in a minute, and she is going to show us in a minute how she uses AI models to understand their behavior, particularly with regards to infection and effectively epidemiology. And she also was a Vico Fellow here in 2013. So the format for the evening is each one of the panelists is going to do a short presentation, sort of 5 minutes or so, present some of their work, and then the four of us are going to have a discussion, and then at some point I will open it up to questions. If you could just get that, that'd be great. It's Darwin.
It's time.
He wants his ants back. And then we'll open it up to the floor. We have rules about chairing sessions. Questions come in the form of single sentences which end with an upwards inflection. That's rule 1. And rule 2, and this is really important, a paper was published in 2015 which showed that in public events at science conferences, if women ask questions first, then 50% on average of the questions come from women. But if men ask questions first, then about 80% of the questions come from men. So when it comes to questions, we'll be taking them from alternating between women and men. So please don't let me down on that regard. Okay, so let's get going. So please could you join me in welcoming Sylvia to the stage first.
Yeah, thank you very much for the introduction. So I'm starting because I'm working on AI in ants. So we start alphabetically. My name is Sylvia Kramer, and I became a bit nostalgic when I realized that I've been here 20 years back, actually 2006 as a junior fellow, and I was really lucky enough to be part of a focus group on evolutionary immunology, and that was in between my postdoc and starting my own group, then back at the University in Regensburg. And when I was here, I got access to the enormous library that Vico also had old papers that you could dig out. And together with Paul Schmidt-Hempel, who was the convener of the group, we then wrote a paper bringing together all the knowledge that there was at the time for what we basically called social immunity, and that is collective and cooperative disease defenses. And this basically review is what came out of my time here, and this has become basically the basis of my work ever since. So what is social immunity? So I study ants to have a look at social immunity, and that means that a colony as a whole is actually providing a lot of hygiene to prevent disease spread and epidemics. And we use garden ants here, and these ants produce formic acid, which many of you know. And we can visualize this formic acid as these red spots, and this is actually very highly antimicrobial. So they disinfect their nest by using their formic acid, their self-produced antimicrobials. They also perform sanitary care to each other. If an individual carries fungal spores on its body, it's groomed off, so the pathogen is removed before the individuals even get sick at all. And we also study social interaction networks of colony members and how they are changing in the light of infection. So we found that ants are also doing social distancing and thereby reducing transmission. So how do we study these social interaction networks? We have students in the group who are able to cut out QR codes of 0.65 millimeters size and glue them on ants. Then it's amazing enough, but you can really purchase systems that are then automatically tracking QR codes on insects, in our case on ants. And what this gives you is that like 2 or 8 times a second, you get the XY coordinates of all the colony members, except the ones that are just bending their abdomen at the moment, but then you get them in the next frame. So what we then get from this information is basically you reduce all the information to these QR codes, and then when 2 QR codes are close to each other, you infer that there has been an interaction between these individuals, and then you can create what is called proximity networks. So one knows that these ants have been interacting and how much and when, but we don't know what the ants are doing. But ants do show a lot of different behaviors, and so we have then been sitting down and trying to just annotate by hand, or as human annotators, just the hygiene behavior of cleaning their own body, providing sanitary care and receiving care of groups of 6 ants in 100 replicates of 2 hours. And 2 people in my lab have been each spending full-time half a year on what you see here as an example. So basically, what you can do is that you then get all the interactions, but you cannot do this at a colony level. And this is what we are doing now by working together with an AI group at our institute. It's a very diverse institute. These computer scientists now help us to really provide a high throughput system to annotate ant behavior. So what you can see in this video is that 3 ants are just here in a small dish. You can track them, and then you can see the behavior. Whenever it's occurring green, the AI model is detecting this, what is called grooming behavior, where one ant cleans off the spores from the other individual. So this helps us to show which treatments are eliciting grooming, and you can see already in this video that you can really then look at a lot of replicates at the same time, and so this is really a possibility for us to get high throughput behavioral annotation, and the accuracy of these models are amazing. So now this is where we stand now, but what we want to do further is that, as is shown here in these QR codes, you can only get really pixelated information on what the ants are doing. And what we have solved now is the upper layer here, the hygiene behavior. But of course, ants are doing a lot of other behavioral interactions, like food sharing or recognition behavior, what is called antenation. And we are now training basically all these other behaviors as well, so that we get the full behavioral repertoire of the whole colonies. By looking at different behaviors, you can also see how versatile different individuals are, how much they engage in this behavior and in that behavior, and how this may change over the course of infection. So like this, you can then disentangle all this proximity information and find out what behaviors are blocking and which are promoting disease transmission in the colony. And this is our supervised or targeted approach, and we are currently also developing some kind of untargeted screening approach, or basically our AI colleagues do that, we don't even have to know which behaviors the ants are doing. Basically, what we do is that we have a group of ants together with their brood, and then we present them whatever type of cue. This can be a smell of disease, etc. And then you can just make the videos, and then you can give them a lot of different social cues. And then we can see which of those smells or cues are making any change, and so we can screen for interesting information that the ants can receive, and then we can go back in and really analyze this by ourselves. So this is basically what we are currently doing and what we hope to do in the future. And this was my part on the ants, and now we go to B for the bees.
Yeah, so cool. Yeah, and it also so very similar to what we do in bees. So I'm a computer scientist, and I didn't really want to work only with computers. And very early on, I was interested in groups and social behavior, because for me, this is a computer. This is a computer, right? So we have brains, and this analogy is very like intuitive, right? So we have processors in our head. Yeah, that's fine. But we do communicate, and we exchange information. And it's still very fascinating to me to understand how exactly we compute and how evolution changed that computation and how AI may affect it, right? But we can't do it in humans, you know, privacy issues and all these things. I don't have the data. And so that's why we use bees, which carry these very similar but not the same tags. Because
What happens if we actually scan one of those QR codes?
these are not QR codes.
Oh, okay, right.
Yeah, these are our own.
It doesn't take you to
No, the phone will say, I don't know.
Oh, okay.
Yeah. So let's go back because, no, I can't go back. Our tags have to be round because they inspect the cells. And if there's just one little edge or a corner sticking out, it's going to fall off. So I'm going to spare you the details about how this has been developed, but we can basically track everyone, the tagged ones and the untagged ones. But the tagged ones now have a name, and then when they go out, and that's the very interesting part, because that's where individuals navigate, where individuals collect information that the others don't have. This is what that then has to be communicated inside the hive. So it's really interesting to see how that information spreads. Very similar to, you know, hygienic behavior or you know, spread of pathogens or so. Information can be treated as such. So obviously we are looking at these networks as well, but more from an information processing point of view. What we also do is robots. So, you know, you can observe a system and still don't learn anything about it because it's just everything is correlated and you don't know what causes what and is it really like we think, right? So we have to test hypotheses. So the left thing is a system that I built in my PhD with Randolph Menzel, who told me he would be here. I don't see you here. I hope you are. No. So this was to test whether or what kind of cues are important in the honeybee waggle dance. And the right system is the RoboFish system that was built with Jens Krause, who is in the back of this room. And the goal of RoboFish is something very different. If you ask different people, you get many, many answers. For me, this is what we have been doing. So the RoboFish basically is a 3D printed replica. We use guppies. I won't shoot you. We have been testing this with other species as well. Then below the tank, there's this two-wheeled cart with a little magnet, and then this couples into the magnet of the replica, and then, yeah. So this is a simulation that we've built to train an AI robot to interact with a model. Could be like a very old, you know, social forces model, could be a very modern machine learning model that basically looks at data, represents the data, and then we train the robot to interact with this model in a way that makes the model, the fish, to go to these target spots. And then we test these in the real world with the RoboFish system. So it's basically saying if the world would work like that, the fish model, you know, this strategy should work in the real world, right? Testing this with the real fish here. That's the real fish, that's the robot. And so this way, this way, we are basically, you know, doing one step of the scientific method, right? So we have a model, we create hypotheses in the form of the robotic behavior. That should work, right? So if I move towards you, you should go away, right? That's the hypothesis. And I could maybe shepherd you to go to the goal. And I won't tell you what the results were because it's very interesting. And we need some people to read it. And maybe,
I'm not going to allow that.
Yeah. What,
we
people
are
to read it?
We are going to talk about
Yeah,
the results.
yeah, okay, we will. Anyway, so, but if you look at what AI has been used for, and if you look at our job, you know, doing science, to observe a system, forming questions, building models, creating hypotheses, testing these hypotheses in experiment, analyze the data that comes out of there, and write down everything. All these things have already been done and assisted by AI. I'm using that every day, many, many hours per day. And so it's very close, at least for me as a computer scientist, to think of the AI scientist that does everything that's on this slide. And at least for the RoboFish system, that's something that I am preparing to do. So, you know, ethics committees will probably have something to say about that too. But sorry, this is your slide. Yeah, but that's what I think we should talk about too. Yeah, thanks.
Okay, so I put some sort of more down than upsides things there because I felt like when Dieter invited me to come here, he probably expected some fabulous, you know, how have I embraced AI in my research as a theoretical evolutionary ecologist. And so far I've actually been quite, you know, call me old-fashioned, call me resistant, call me stupid, whatever. But given that we are talking about A, ants, B, bees, and C, what is it? I mean, some sort of concepts, collaboration, or, you know, you name it. But I'll start with the collaboration / creativity aspect. Creativity also begins with a C, so this works. So here's, I've been preparing these slides on how I write papers, and here's me, one of them, celebrating a paper acceptance with a co-author. And I've had a lot of co-authors over the years, and I've really enjoyed these kind of things where there's, let's say, just two of us, and this other person has complementary expertise to me, that also begins with a C. And we bring in some viewpoints that we couldn't have done together, couldn't have done alone. And then you can ask, you know, what happens if a third person comes along? And often, actually, despite what seems to be kind of like a detrimental interaction there, this can also be very much fun. But then there's also these kind of papers. So you see a lot of people in the background. And just to explain, this is actually also a dear co-organizer, Ed Iverme Cook. We organized an evolution of aging workshop in Ingelheim near Mainz last year. And we had fabulous people, we had a lot of fun, but we decided not to make everybody write a massive group collective paper together. And especially I was a bit against that idea because I've been involved in those kind of things and they tend to become a bit bland. It's kind of like a paper written by committee. All the perspectives have to be there and everybody just gets like 3 sentences of whatever favorite topic, and I've read so many of them, and we encouraged people to collaborate, form new things with 2 or 3 people, whatever, and these kind of things have happened, and I'm actually quite happy about the decision. What does this have to do with AI? It's this paper that I then found somewhere, and this is not about using AI in these sensible ways that we want to know where the ants go and what the bees are up to and all that, but this is about creativity, getting ideas, and you can see, you can read the title. Just to tell you what was going on in here, I don't actually know these authors, but this is sort of people who try to work as, or try to learn writing skills, fiction writing. And they were either allowed or not allowed to use AI to create ideas. And individually, they somehow objectively then rated how novel their ideas were. Individually, they got a benefit. But when you look at the entire group, they were kind of like spooning from the same soup. So then collectively, the non-AI group actually did better, which I think is interesting. Well, I'm not a fiction writer. I'm not even an aspiring fiction writer. But I like to, so my work is about trying to find out some sort of underlying processes that can explain biological data. And well, I don't really want to compare myself with Newton, but but you know, this is something where Newton was somebody who was really thinking outside the current box back then. People had built more complicated models about planetary movement, because if you look at them from the Earth, then there's all kinds of interesting loops happening here and there, and then you can put all kinds of circles, circling around circles and all that. But he figured out that the real solution is astonishingly simple, and you just need a change of perspective that nobody had thought of before, and then you can explain everything. Now this paper, which is a recent International Conference on Machine Learning paper, and I have to credit Jussi Lehtonen, who is a fantastic former PhD student of mine, who these days does modeling on why do males and females evolve to be different, and has taken that field. You know, Peter Hammerstein would love, you need to read all his papers, they're great. But he told me about this one, where they had now put machine learning as one form of AI, and given it a lot of data on how planets are in different locations. And you can see that it finds that, you know, we can find the circle, but this is the equation they get. And this, of course, is completely non-generalizable to the next planet, because it doesn't capture the underlying physics, but it fits the data. So these are the sort of worries that I tell students: don't use your AI naively. Use it. I mean, I'm not going to say that, you know, this is bad, should be banned, but be aware of the downsides. And because I sort of like to collect these kind of things just to create a bit of discussion here, here's a truly fantastic one. And here I'm sort of moving to the large language model world. Some people are already laughing because you see this. So you know this famous illusion where there's two lines and one of them has the arrows this way and the other one that way, and then we think that, you know, they are different lengths even though they're the same length. And if you ask, I don't know which version of AI was asked here, but this is saying that, well, actually this is the illusion, da, 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 da. And this just teaches you how little reality goes into it. This is the reason why it produces this answer, is that the only context that humans talk about these lines is when the illusion is being exemplified. So remember the dangers of talking to an AI that is just listening to what people say. And here's another one. You don't maybe have time to read the whole thing, but people say online all kinds of things. Sometimes they willfully want to make you believe something. Sometimes they make a satirical comment. So here's somebody was, the Beaverstone, that's a satirical magazine published somewhere, and somebody said that, you know, these particular people somewhere in Newfoundland, they want to separate themselves so much that they invent this time zone that is 12 minutes annoyingly away from the others. And it's satire, but it gets picked up by large language models, and suddenly this is what students find if you do this. And I want to go back, you know, I started with Newton, and if you go back to medieval times, and I'm a bit nervous here because I'm not a historian of science, but then my final two slides, they are actually from a course that I was teaching some years ago, before AI became such a huge thing in biology. And the class was kind of, I called it boldly, how does science produce knowledge? And I wanted to go through some famous examples of how did we figure out that vitamin C deficiency is actually the cause of scurvy, and all those kind of like old things where we really know the answer today, and how did we get there. And in the very first lecture of that class, I showed people this old text. It's written in very old-fashioned English, but basically the story is that there's this monastery, and they had a fight about how many teeth does a horse have. And the monks couldn't agree, and then came a young monk who very naively said that, well, why don't we look at the horse and open the mouth and count? And then, of course, all the other monks were outraged that, you know, you have to know that, you know, you have to go back to Aristotle and, you know, all these texts. And as far as I know, if I've read my history books correctly, it's suspected that Francis Bacon was the author of this story. Nobody seems to know for sure. Please tell me if there's more info on this. But Bacon, I'll just end with this slide, which also unedited is from that course I was teaching, because he said that clearly he was of the opinion that we could actually ask nature instead of just trusting what somebody else has been saying. And he came up with a method of saying that you should make lists of all the things where your favorite phenomenon occurs, and you should make a list of all these cases and instances where it does not occur. and then you rank your lists according to the degree, if there's some partial information in there, and then you should be able to deduce what matches the occurrence of the phenomenon in one list and not in the other. And you can sort of see that this is sort of correlational analysis of some kind, and some details in there, and this is, he was saying, this is how we arrive at causalities. And of course, this, as you can imagine, this was just the start of the course, because I'm inviting people to think, why is this not complete yet? And I think I'll kind of end there, because I want to leave it a bit open-ended for discussion. However, I want, because Sylvia started by saying how wonderful it was to be at Vico. I was here in 2014, and I think it's forever unfair that I was here for only 2 months, but it's counted as a complete fellowship. So I just wanted, this is a good stage to say that. There you go.
Okay, we have a lot to talk about.
Oh, I promised, I had one job. Yeah.
So you mentioned Aristotle, in Animalia, which is a brilliant, interesting book that we don't really need to refer to anymore, he does state that women have fewer teeth than men, which is a question that also could have been answered by counting them.
Yeah.
That wasn't the main point of your talk, but I thought I'd comment that. I want to do some definitions first, though, because this is a public-facing audience. You guys are, some of you are scientists, but many of you aren't. And we quite casually use certain words that aren't necessarily used in the same way that normal people use them. So, Tim, what's a model?
Like, from what kind of point of view? From the AI point of view?
So we talk about models in science all the time.
Yeah.
But what does that actually mean in terms of answering a question about science?
If I talk about models with my students, we 100% of the time mean the computer science machine learning models, which are basically learning a mapping between input-output spaces, fitting the data. And obviously, we even observing the world ourselves, we create a model of how things work, right? So an imperfect, simplified, useful version of
useful is
reality.
Interesting in that, because there's a famous phrase by the statistician George Box, which is that all models are wrong, but some of them are useful.
I agree.
Yeah. So how can you tell if your model is useful rather than simply wrong?
Yeah, it depends on what you want to do with the model, right? So what do we want to do with the model? We wanted to allow predictions, right? In the end, these are interesting because we can test those. So, building a model just for the sake of the model sometimes happens, but most of the time happens because we want to use it for science. Right? Run it, create predictions.
And match it to the real world.
Match it to the real world.
Yeah, because when it is matched, your work is about matching it to the real world, because then the model is more useful than if it's just hypothetical. I think I'm going to ask Hannah this question.
I was expecting that.
Yeah. What is AI?
Oh.
The reason I ask this is because there's a lot of bullshit talked about AI, and often in the real world when people are talking about AI, they're actually talking about Excel spreadsheets or some quite sophisticated calculations. And you've mentioned we've fused a bunch of AI together in the 3 talks, generative AI and other computational systems. How broad can we be when we're talking about what AI actually is?
So I'm not a computer scientist, so I don't know the formal definition actually. If I think about my experience of when people use it and I'm deducing it from there, I would say that it's any computer-assisted model that is a bit too complex for us to think through all the, from the input to the output. There's a certain black boxness in there.
And Tim, what is a model? But it is interesting to think about it in those terms, because we deploy these technologies, some of which are simply bought, that you can buy, and I think, Sylvia, the tools that you're using they're modified but off the shelf.
Well, I mean, there have been some scientists who developed this, and you developed your own system, and other people have developed them. But nowadays, yeah, you can basically purchase them and employ them for people like
If
me.
you need
who
one,
don't
I sell you
want to
one.
It's for a while I've been thinking that AI, in its most well understood and most used form, it was created as a tool that didn't have a function, and that we've spent many years trying to create functions for the tools that we've already built. I'm not leveling this at you as scientists, but that feels like why there's so much problematic AI in the real world. Is that fair, do you think, Hannah? I mean, the example you gave of the fake news, a really significant real-world example of this is in Robert Kennedy Jr.'s health reports, which contained 29 references, academic references, which were made up. They did not exist.
I mean, my favorite example of that kind is that there was an email in my inbox, somebody wants to join my lab, and said that I was so impressed by this paper that you wrote last year, title, and you know, how inspirational, insightful, and all that. And the title was something that I feel I could have written, but I didn't write that paper. And then you feel sorry for these students who fall into these traps and think it saves them time and impresses me. And this is why I teach people to be a little bit careful and do a bit of fact checking.
Yeah. It was interesting, something that Dieter said at the beginning, which is that AI is enabling us to ask new questions. So not just tackle questions that we know exist, but this relates to the question of creativity, in which I think, Tim, you were talking about. Is AI capable of creativity in asking scientific questions that we haven't thought of yet? Or is it simply crunching data and presenting new avenues of pursuit for us? I think that's both Sylvia and Tim can answer that.
I mean, my postdoc told me that when ChatGPT came out, he basically said he typed in what would be Sylvia Kremer's next paper. I've never done it. I don't know. I make up my own research, but yeah. So basically, they can bring together your past and maybe something that is out there in the field, maybe open questions. And I guess some people really use it to create also some ideas.
Tim, do you think it can create new ideas?
Yeah. I think it has been shown that in some instances there were emergent properties, emergent capabilities. And in some other cases where we thought this is a generalizable kind of tool now, it has been shown that basically all the data was learned by heart. So it's just replicating data it has seen. Mm-hmm. So it cannot apply the rule in the data to new unseen data. So both happens. We are surprised that things work, and then we're surprised that things don't work. And we don't understand. I think this sums it up.
Well, that's Hannah's black box, isn't it? So there's input and output, but we don't necessarily understand what's happening in the middle, which feels like that should be problematic for scientists.
And can I answer this? You asked only these two people, but I could also provide something, because coming back to my first slide with the shadows and the people, and I think that think about the sort of two large groups of people and the blandness that follows from there. That if I wanted to write my next grant and actually ask AI, I would try to limit the input somehow, and instead ask kind of like, if Sylvia and I wrote a grant together, what would we do? Or Charles Darwin and I, or something like that who we cannot invite to BICO anymore. So the sort of limiting, paradoxically, could actually create more creative solutions than the what if we had all the data of the world, when you end up more in the middle.
Do you think, so if you think of AI as a tool, a really sophisticated tool for doing sort of intellectual heavy lifting and numerical heavy lifting that we're simply not capable of doing, does that allow it to be creative? Because tools are not inherently creative. They rely on us inputting creativity to use those tools effectively. I'm going to talk about the science a bit more specifically in a minute, but I think these big ideas are important.
So maybe not exactly answering the question, but what I found really interesting is that also our computer scientist colleagues that don't know what is basically taken into account, what is learned by these models. We know, we look at the behavior, we say this is this behavior, but we have no idea what the model is actually learning there. So when we asked our model, out of 20,000 different things, what is significantly different between our two treatments? One was the grooming behavior, and the other one was the adding lines that we had put around to place our Petri dishes on the right place. So these two were the outcome of these models. So one of them made a lot of biological sense, and the other one was really just by random that some of the ants on the left upper corner where this adding line was were grooming more over all our replicates, right? So call this creative because it doesn't make any sense, right? But you really have to evaluate those things very harshly, right, to make sense out of it.
Is prediction, they're prediction machines, right? So they're large language models, and machine learning is effectively very complex prediction machines, which is why ChatGPT needs the work of authors. It's probably scanned all of our work in order to predict what the most likely next thing is going to be in a sentence. And it's very impressive that it can do that. Yep. But if it's predicting, if it's sending itself off in directions which are nonsensical, like in that example, is that inherent to the system, or is it something that it will get better at?
So I think that we, as a behavioral ecologist, if we train models without the knowledge somehow what you have to be careful with, then you're creating more and more wrong models. And this is why I think it's actually very important to work together with specialists who try to understand, or our colleagues, they really try to find out where biases occur. So they help us train unbiased models so that we can really use them. But I would not even have known that also AI has bias when looking at videos of ants, right?
Explain what you mean by that. How does it have bias in the system?
Yeah, bias means that they are taking in things that don't make sense. You can maybe explain this a bit better, but where we position our Petri dishes, etc., and basically coincidences can be taken into account and are wrongly learned as something important. Yeah.
Let's say you have 100 photos, and 99 of those photos show elephants, and one photo shows an ant. If you train a model to predict what is on the photo, you can have a simple lazy model just saying it's an elephant. It will be wrong once. High accuracy, right? And so that happens all the time. So and we do know how to understand what a model has learned. It's a lot of work because it's such a huge thing. And right, so it's not like our brains. We don't know the activity of all our brain cells. We don't. For an AI, we could, because we can access all these neurons. We still don't know what that means. And even if we would know the activity of all our brain cells at a given time, we wouldn't know what that means either. So yes, we can find out, and we can test and identify correlations with what's in the data. It's a lot of work. It's a lot of time and effort that goes into this. And then we can find out what this model has learned, what it represents internally. The thing that is really frustrating is we cannot control what it will learn in the training process, right?
So does that mean that AI is incapable of serendipitous scientific findings? The stuff that we spend so much of our time making an observation and thinking, oh, that's interesting, and then it sets you off in a different path.
Depends on the data and it depends on the training objective, I would say. I think it's definitely possible. It's just, these are vector spaces, right? Data lives in a vector space, blah, 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 and then, the internal representations represented inside the model, contain all these data point clouds. And then there's, in the output, some mapping it learns. And the ML, machine learning designer, designs these spaces, and then it just throws a lot of data on this model, and then it learns to represent this data. And that's what we do. Very blindly throwing data at it and trying to figure out what the best input-output space is. So you mentioned predict the next word in the sequence. Very easy. There's a model saying, a paper saying, increasing the model size is very simple, but it's very effective. And that's 10, 12 years ago, and it has been implemented and reiterated all the time because it works. You just increase the model size, you just increase the amount of data that you throw at it at this simple task, and it's marvelous what these models can do with this simple objective.
I mean, it works so well that they had to steal all of our work in order to build those models. But that's a different political point. We won that first case, by the way. Hannah, you're the sort of skeptic on the panel. What do you think about that? Not the stealing.
No, exactly. Which bit of the last 5 minutes?
The notion that this is basically a recapitulation of the scientific method, but it's dependent on what you're putting into it.
Well, of course, not only on what you put into it, but also on how you define the question. Like in preparation for this event, and of course I had no idea what sort of questions you will ask me, I was looking at kind of like very nice cases of AI use that have been in the media just in the last couple of weeks. And one of them, for example, was a breast cancer study. I don't know, many of you have maybe seen that because it was in newspapers, where you could choose, well, the patient couldn't choose, but methodologically what they did was that there might be two doctors making an expert opinion, what's wrong with this woman and should this kind of treatment be applied or the other one, or is it even breast cancer or not? And then they had an AI opinion, and the AI was much better at detecting those cases that are very hard to detect. And if I am ever in a situation where AI detects my breast cancer, I will be, cancel all the bad things that I ever said about it. But it's interesting because it's a very well-defined problem. We basically know kind of roughly what to look for, but it's just a very hard task in each individual instance. Whereas if I'm thinking about it kind of like, figure out that the planets actually do something that nobody until then realized that you could look at it from a completely different angle, it seems to be much harder in those kind of cases. And I guess me as a scientist, the reason I end up being to some extent less enthusiastic than, for example, Sylvia, is that I work on trying to create the simplest possible representation of something that can explain not every detail of the data, not every single breast cancer case, but something about the underlying logic of why does evolution go this way in bigger species and that way in smaller ones. So it's a bit of a difference in the why question.
So it's like you're trying to extract like a single strand of information about a complex system, whereas AI is enabling Sylvia to model not just multiple strands, but also potentially a whole colony of ants rather than just 3. Is that fair?
Well, I see my use as a really more like a user case for high throughput, right? That we expect the AI to do exactly what we could do, but just in much, much higher volume and speed, which then allows us to answer questions that we could simply not do in our lifetime, right? So this is this targeted approach that we have been using. So I really see it as a tamed model, basically, that does exactly what we want it to do and as good as it can, which is pretty good, actually, in accuracy. I have not tried out this, what we call this untargeted approach yet, where it's basically like a screening approach, that you say you have a couple of ants and they do whatever type of behavior, and we have no idea what these ants are doing and whether they react to a specific cue at all. And then the AI would give us, for example, they have like 20,000 neurons, and then they would also just give us pictures and say, this is different. in this approach. But then we have to make sense out of it, right? Because then we see those pictures, and some of them for us say, okay, we can see it, that this is this behavior. But in others, we have no clue whatsoever. And then I see it as our task as a researcher, then again, to really find out what would this particular smell potentially induce in the ants, and what can we expect, and so on. So I think I'm happy to have my own questions and not ask AI what should I research next, and I rather use it as a tool to be able to create these data.
But you still have to have you have to have a human intervention to scrutinize what the output, what the AI is suggesting. That's a big part of it.
Yeah, so basically we as a human annotator first give it what is called a ground truth. So we analyze the videos and tell them in this frame, in this picture, the ants are doing this behavior, and in this they are not doing it. And then they learn it exactly from this, but we don't know what type in the picture the AI learns to infer that this is the behavior.
In my very first scientific project when I was an undergraduate, I was measuring the wingspan of a particular type of fly, and it was a summer project, and the flies were all frozen. We'd starved them to see if they were going to be, if their wings were asymmetric, and I spent a summer measuring these flies' wings. And when we did the analysis, there was this huge left-hand bias. So like a really significant amount of the flies were significantly left-handed, or their wings were bigger on the left-hand side. It was so significant that we didn't think it was true at all. And it turned out the lens had a bubble in it. The microscope, which cost £20,000, had a huge bubble in the lens. And so that was, when you were talking about that just then, it really felt like, you know, if this result is true, this is major. This is going in nature. It wasn't true. There was a bubble in the lens.
Flies were flying circles.
Well, they were. Yeah. That is also true. They were terrible flies. Actually, Sylvia, I want to ask you a question about the research itself. So we've talked about sort of concepts, bigger concepts about AI. But am I right in understanding that the thing you don't really, the question you're asking, because this is the unknown thing, is the transmission of infection. You don't know whether it's as a result of grooming or as a result of proximity, and you're asking in the experimental situation, you're asking the AI to work out which behavior is which. Is that roughly right?
No.
Excellent.
So at the moment, we are only limited to know, for example, when two ants are close together, or we know how much time they spend with each other, and at the end of the period that we are observing, we measure the load of the pathogen that is transmitted from this, say, 1 or 5 colony members that carry the infection in the beginning towards all the other colony members. And then we kind of try to correlate the pathogen load that each individual has received to the interaction with the infected individuals. And so normally what you think is that there is a high risk of infection when you're interacting a lot. But when this interaction is actually hygiene behavior, where others come and remove the pathogen from you, a high rate of interaction should actually reduce the transfer between them. But then on the other hand, grooming somebody who is infectious to remove the pathogen is a risky task for the helper. So we are now finding out via like fluorescence methods and really observing the behavior that there is a lot of pathogen transfer towards the helper, but that they can get rid of it again because they clean themselves very well. So we actually haven't really understood whether this grooming behavior actually helps transmitting the pathogen to other colony members, or whether it's rather just passing by each other. So what we want to do is we want to look at disease transmission and also model the spread when you just look at proximity or just look at grooming or at all the different behaviors. So to understand what is actually the driver of infection transmission or reduction. But then it's actually also interesting that disease transmission is not the same as pathogen transmission, because you only develop the disease when you, for example, get a high load of pathogen. But if you only get 1 or 2, for example, fungal spores, you can actually boost your immune system. And then having some pathogen transmission to other colony members can actually lead to some herd immunity. So it's actually a relatively complex system, and there are a lot of different factors, not only including the interaction, social interaction networks, but also the individual immunity that is included, and the virulence of the pathogen, and we try to look at that whole picture. And this helps us.
So we study these systems because they're inherently interesting, and we do not need to justify that type of basic research with practical applications. But as you were talking there, I was just thinking, this surely is an incredibly useful model for epidemiological disease transmission for, I don't know, some kind of airborne infection maybe that humans might be vulnerable to? So airborne, I'm not sure, but so I think one really big benefit is, as Tim has said, those social insects are systems, they are whole systems on your desk. So you can look at a whole group of interacting individuals, and the thing is you could observe all the interactions and you can observe all the pathogen spread. And by this you can then really create mathematical models that help you predict the epidemiology in those systems. And I think you can then kind of go via this abstract way that you then have better models than you would be able to generate if you only see half of the situation. And those models, they can then apply also to other disease systems, some of them at least, that have similar transmission dynamics. And Tim, what is a model again? No, I'm joking. You don't have to answer that. Presumably, this is the way you think about this as well. There must be like.
It's very similar. Yeah.
So, like real practical implications. Do you see the transfer? Do you see knowledge transfer from this kind of basic research into human epidemiological studies, or just modeling crowd behaviors?
Oh, yeah. So one of my PhD students, he created a model, like an AI model, a machine learning model that would basically factorize a matrix. So the social interactions.
No. I will not allow that.
Hold on. 2 steps. Social interactions are basically, you can write down as a matrix. Yeah. Who is interacting how much with whom. And then what we think is that every individual has a latent, unobservable kind of state that makes it interact in a certain pattern. And so we could learn a model that would predict each individual's internal state that would minimize the discrepancy of the predicted interactions and the observed interactions. And why do I tell you that? Well, because we applied that to human health records. It's the same thing. You can apply the same system to recommender systems. So which user is interacting with what movie or book or other user in social, like in human social networks or Amazon bookstores or whatever. So if the underlying data structure is the same, and physicists and more mathy people would not see the discrepancy of what I'm just saying, it's the same. It's the same. And so it's a huge transferability, not just in social networks, it's into all networks, if you work in method development.
Hannah, I couldn't tell whether you were looking skeptical or agreeing.
No, I agree.
Yeah. Shall I wrap this up so we can go and have a glass of wine? Okay, thank you. This was super interesting, and we've had a complete range of views. I feel like we've been bullying Tim a little bit, but he seems to be able to take it.
That was my role, wasn't it?
I think, well, I didn't cast this. But could I just ask you, before we go and have a glass of wine, to thank my three panelists, Hannah, Tim, and Sylvia.